evening. We are starting the Translational Longevity series of presentations, and our first presenter will be Dr. Vijay Padu, who is a senior scientist at Renix Life Sciences, and he leads the bioinformatics, genomics, and next generation sequencing research related activities in our organization. He's currently working on the neoantigen cancer vaccine pipeline projects here. Vijay holds a PhD in life sciences from the Homi Bhabha National Institute, Navi Mumbai, and his PhD studies focused on cancer genomics. And previously, Vijay has also worked as an assistant professor at graduate and postgraduate degree colleges as well. At Renix, he has gained experience in scientific, operational, as well as administrative activities. Over to Vijay. Over to you, Vijay.
Thank you, Dr. Divya, for that introduction. Welcome, everyone. For our Translational Longevity series, I have taken the topic of genomics. Can it answer longevity questions? So this topic is of interest because genomics is a newly rising field, and which can give a comprehensive information about the genetic blueprint of an individual. And in that scenario, can we predict a person's longevity based on that genetic blueprint? That is the question that I'd like to tackle in this particular presentation. So these are the references that I have selected for today's presentation. So coming to introduction. Over the last 2 centuries, we have witnessed increased life expectancy worldwide, but still it is highly variable between the countries. Everybody wants to live long and fully satisfied life, but there are different factors that come into play in determining the longevity of individual. Of course, for this subject of today's, that is genomics, we will try to see whether genomics can answer the longevity questions. Can it be used to predict the longevity of individual? So, with the advancement of technology and social progress in different countries in a variable rate, we have seen increase in life expectancy in the last 2 centuries. The reasons for this extension has been because of improvement in the healthcare to the citizens, and improvement in the hygiene and improvement in the nutrition. All this because of all the technological progress that we have made in the last 2 centuries. But even if lifespan has increased, healthy life expectancy, that is the health span of an individual, has not increased at the same rate. Now, what are the factors for that? That we'll see in today's literature. So age is a main risk factor for majority of common diseases that contribute to the disability. As the person ages, their organ systems start to fail and function at a reduced rate, and therefore individual cannot retain its abilities once he had in its youth. So aging process is of very much interest for us biologically as well as socially also, because in future there will be increase in the number of individuals who will be aging and will need age time care. So reaching an old age does not necessarily result in higher degree of age-related disability in long-lived individuals from families expressing exceptional longevity that may reach high ages without major disabilities. So this is the thing that intrigues scientific mind, that whether there is any underlying genetic component to the longevity, that whether these exceptionally long living individuals have some genetic components that are leading to their longevity, and can we identify them? So a variety of different population genomic studies have been conducted, and they have given us some insight into this. So Moro and Dazolti twin studies, they have revealed that genetic contribution to the human lifespan is 25 to 30%. So this is a lot contribution into the lifespan of an individual, and this genetic contribution is mainly apparent after age of 60 years and later. Of course, excluding from early days due to a variety of different reasons. So once a person crosses this particular age threshold, then this becomes more apparent in the highly aged individuals. The genome of long-lived individuals is investigated to identify variants that promote healthy aging and protect against age-related diseases. Of course, these individuals who are living 100 years or more they are ideal candidates to identify or look at their genomes, like what particular genetic factors they have that are leading to their exceptional longevity. So these research papers have also identified such genetic factors that are strongly correlated with longevity. And longevity can be affected by presence of protective genetic factors against age-related phenotypes and diseases. And these genetic factors are needed to be identified, not only contributing to longevity, but those factors that can protect against age-related degeneration also, and phenotype and diseases that occur due to the old age. So are there any genetic factors that protect against these things that can also be identified through population genomic studies? The present challenges right now are to identify whether genetic components are the main factors or the lifestyle plays the major role in determining the longevity of individual. Of course, both have their own contribution, but which one is the primary factor? Is the genetics determines the longevity, or lifestyle has a major contribution in determining that? And also which one outweighs the other and why? Which has the upper hand? Even if a person is born with good genetics, does bad lifestyle will lead to early death of that individual? Very probably yes, but we need to study it scientifically and answer it so that we have definitive answer for that. And are genetics and lifestyle complementary? That also needs to be answered by population genomics level studies or some case control studies also. And genetic predisposition to the lifestyle diseases. Are there certain genetic factors that make an individual predisposed to a particular lifestyle diseases such as diabetes or heart arrhythmia or blood pressure, high blood pressure, etc. So these are the present challenges to be studied in this field. Now here in this table, it is apparent that as we have progressed through the years in the last 2 centuries, the lifespan of male and female has consistently increased, reaching up to 79 years in 2011 for male and 84 years for female. In these different studies, they have tried to identify in long-lived individuals what are the genetic factors that are highly associated with their longer age or longevity. And these are the list of genes that have been found to have high correlation, statistical correlation with these high longer living individuals. And these include apolipoprotein E, ApoC3, ChRNA35, LPA, HLA-DQA1, DRB1, SH2B3, CETP, and HTERT. But still, there is an issue with these genetic studies, because if one particular study finds a particular gene locus to be highly correlated with longevity, but another study would not be able to replicate that. So lack of replication in these particular results is the main issue to determine what are the contributing factors these particular gene loci are playing. So as described in this particular karyotype, there are different locus that have been identified to be highly correlated with the longevity. But unfortunately, these have not been replicated into the other studies. If they had been, then there would be very strong reason to believe that these particular locus are contributing to longevity in an individual. So the question comes, is genetics a sole determinant of longevity or not? Because there are other factors that work along with genetics, because not every genetic variant that is present in an individual will necessarily reflect in that person's phenotype. So there are certain processes such as epistasis, epigenetic effects, and gene environment interactions that can play a role into whether a particular allele will express or not in an individual. So these controls, these are about the genetic level, at the transcriptional level, whether that particular gene allele will get transcribed or not. That will be determined at this particular level. As an individual has 2 copies of a particular gene and alleles from either parent, mother and father. So which copy will be expressed, either both will be expressed or any one of them will be expressed, it will be decided by these processes. So is the genetics a sole determinant, or there are these processes also that will govern the expression of these particular genes? That also needs to be studied. Here in this particular table, we can see that the improvement in the standard of living has resulted in increase in the lifespan. And this particular table shows us the potential biomarkers which have been shown to be associated with the mortality in prospective studies of different ages. And this association has been found in different age group after certain age group. So, such as blood pressure, diastolic and systolic, body mass index, cholesterol levels, cortisol levels, reactive protein levels, creatinine, and fasting glucose, gait speed, grip strength, etc. All of these, they seem to have a role to play on the longevity of an individual. And these have been found to be significant in these studies. So to summarize the all the research articles and idea of this presentation, so a definitive causative association of genetic variance and longevity has not yet been identified. Even if they have been identified, they have not been replicated in other studies. So this is where it is lacking, that efficacy of genomics or genomic tests, whether they will be helpful right now in identifying the longevity, potential longevity of an individual. So what will be the use of genomic testing? Right now, there is a prominent role of genomic testing, and whole genome testing is getting, sequencing is getting prominence as a major avenue for identifying different genetic disorders and predisposing factors for different genetic diseases and disorders. And what will be the role of those particular genomic testing? So it indirectly, we can use that particular data to identify whether that individual has some risk factors in that genome, which potentially may arise in the lifetime of that particular individual, and may contribute to the reduced lifespan of that particular individual. And if we have that particular knowledge that this particular individual will develop hypertension going forward, or will present with diabetes, then some preventive measures can be suggested to that individual, so that that individual will make lifestyle changes and avoid severity of those particular predisposing factors that will arise in that particular individual's lifetime. So in this way, this information, genomic testing or genomic sequencing, will be useful. And another aspect is, as genome sequencing is gaining in popularity, so these studies can be performed at the population level, and in the long term, we can identify whether there are indeed some regions in the genome which are associated with the longevity of an individual, and which can also be validated in different populations also. So in this way, we can utilize genomics in the field of predicting longevity, right? So thanks for listening patiently. If you have any questions, welcome.
I don't see any questions from the audience. So thank you, Vijay, for this informative talk on genomics and longevity.
Thank you, everyone. Thank you for patiently listening.